It's $5 or free for students. So students don't have to. Let me get this. There we go. That's the streaming right there. Okay. All right. So I'm Jason Heindel. Thanks for coming to our first monthly meeting of this academic year. And we're hosting Nicole Marino, who's present in front here. And before we get there, and before I introduce her properly, let me just give you a couple over here. A couple points about, there we go, about ASM and the branch. So we are the Eastern Pennsylvania branch of ASM. We're one of the oldest branches, and we're definitely one of the most active branches in ASM. And if you wanted to join ASM, if you're not already a member of either the branch or ASM National, it's very easy to do so. Go to asm.org, and then you can join National. It's relatively inexpensive for students. The costs are listed right there. For a branch, just if you want just to become a member of this branch, for students, it's free. So you can do that, and you don't have to join ASM National to do that. You can be a member of the branch and not national, but I might as well do both. If you want to join, they do work on a calendar year, so January to December. And I think if you go now, they will kind of grandfather you into the next calendar year. If that's not the case, then wait until December to join, so you can get the full year benefit. We do have a number of upcoming events that I want to highlight. First off, this Friday is the full clinical microbiology symposium. If you want more information about that, you can go to the branch website, and there's plenty of info there to register, or you can email anyone involved with it. 1 month from now, on October 29th, we are hosting Peggy Cotter from University of North Carolina. That's she's going to give a talk on her research. And if you've heard of the Peggy Cotter Travel Awards for going to ASM Microbe, this is the namesake for the Travel Award. So that should be a great talk. In November 11th, the Norman Willett Memorial Lecture, hosted by the Education Committee, is being held. And that is going to be Jennifer Stanford, who's at Drexel. And it's kind of an STEM education focused talk. So if you're interested in that, please come. And then December 2nd is the PIF, the Philadelphia Infection and Immunity Forum. And that's a full day scientific forum sponsored by the student group. And so you can check out the website once that gets updated with that. I know they've got some exciting potential invites. I don't know where that stands. Maybe I'll get an update later. And our website is being updated, FYI. So if things don't work out great, it's because we're updating the website. But I think it works great right now, actually. All right. Now, it's my pleasure to introduce Nicole Marino to you. So she is a relatively new faculty member at the Department of Pathobiology at Penn. And I do have her own publication of how she describes herself on her website, or someone wrote this. I'm assuming it was you. Okay. So quick background on Nicole and her training. So she took her BA at Rice University in Houston. She had a double major in biochemistry and cell biology. That's not the double major. And classical studies. That's the second component of the double, which I think is pretty cool. And then she took her PhD from Stanford University, working with John Boothroyd on Toxoplasma gondii, which is, if you know anything the Boothroyd Labs does, is quite well done, and Toxos, a fascinating parasite. So if you don't know Toxo, get to know it. And then she did her postdoctoral fellowship with Joseph Bondi Dennemy's lab at UCSF, working on phage. Is that a quick summary? Yeah. And so currently she is an assistant professor, as I said, in the Department of Pathobiology at Penn, Vet. And I'm not going to say what she likes to do outside of lab, but she does have a history of solid NIH funding. Her graduate work was funded with an F31 fellowship. Her postdoc was funded with an F32 fellowship. And then she transitioned out of K99R00. So hopefully she'll continue that. The story she's going to tell us is really fascinating. So I'll let her tell us about translation-dependent downregulation of Cas12a mRNA by an anti-CRISPR protein. So please welcome Nicole.
Thank you so much. Let me just start by saying how excited I am to be here and to be part of this larger research community. And today I'm going to talk mostly about my postdoctoral research, but I'm happy to say that my lab is up and running. We are getting some results. And so I'll share some of those with you today as well. And I always like to begin with this electron micrograph image. of a bacterium that's just being overwhelmed by phages, because it underscores the pressure that phage put bacteria under. Phages are thought to be the most abundant biological enemies on the planet. They're thought to outnumber bacteria 10 to 1. And they're major drivers both of bacterial mortality and also gene flow, and that has a lot to do with how phage infect bacteria. So when a phage injects its genome into the cell, it typically undergoes one of two different pathways. So in one case, it may immediately hijack the host cell machinery and go on to replicate itself and lyse the cell. And we refer to this as the lytic cycle. It's a form of absolute parasitism where the phage is really just using the bacterial host for its replication and its transmission. Alternatively, phages can instead forego immediate replication and instead integrate into the bacterial genome as a prophage. And this prophage can replicate along with the bacterium indefinitely. And in many ways, it's a more complex relationship because the phage is the phage and its bacterial host are intertwined. So it's more of a symbiosis. But note that in response to various cues or even spontaneously, this phage can revert back to the lytic cycle and kill its host. So it still poses a threat. And so bacteria have evolved many different defense systems that target phage for destruction. And some of these will target the phage directly, oftentimes by cleaving its genome. And one example that I'll talk about a lot today is Cas12a. Alternatively, some defense systems instead act by abortive infection, which is a more indirect mechanism for phage control, where the defense system will detect phage infection and then activate an antiviral response that oftentimes leads to a bacterial cell dormancy or death. Which might seem a little drastic, right? Because these are unicellular organisms. And yet it's thought that by limiting the phage infection and preventing it from lysing the cell, it basically prevents the phage from spreading throughout the population. And so it's a form of altruistic suicide. So phage and bacteria have been locked in genetic conflict for almost 4 billion years. And yet to look at this as simply an antagonistic relationship, I think is overly simplistic. Because again, phage are major drivers of gene flow in bacteria, and they can oftentimes help bacteria adapt to the ever-changing environments that they find themselves in. And there's lots of examples in which phage can actually cooperate with their bacterial host. And one example that I always like to bring up is cholera toxin, right? So this is a phage-derived toxin that dramatically impacts Vibrio cholera pathogenesis and transmission, which also then helps both the bacterium and its phage. And Paul Bollenke's lab showed that phages can also promote antiviral immunity in the mammalian host that compromises antibacterial immunity. And that again can allow the bacterium and its prophage to persist. And so there's lots of examples in which the phage and bacteria can cooperate and have complex tripartite interactions with the mammalian host. And so I would argue that to really understand bacterial physiology and to understand bacterial infection, we really have to understand how phages fit into all this. But there's also pragmatic reasons to study this. So antimicrobial resistance, of course, has been a big problem for a long time. In a lot of ways, it's just getting worse. We've come to appreciate that finding new antibiotics is hard. And at the moment, AMR is thought to kill over 1 million people each year and contribute to many more deaths besides. And I saw one estimate recently that it may kill up to 40 million people by 2050. In principle, it could return us back to the pre-antibiotic era. And phages as a therapy actually predate antibiotics. Of course, antibiotics have taken over as the preferred therapy, especially in the West, in part because they are more reliable, they are more broad spectrum. But there is renewed interest in using phages to treat bacterial infection for a few reasons, right? So on one hand, you can often, not often, but let's say for bacterial infections that have resistance to multiple antibiotics, you can treat with a phage and actually clear that infection. And there have been a few cases now of compassionate use where phage therapy is able to treat an infection that is otherwise resistant to antibiotics. But people are also interested in using phages in tandem with antibiotics, right? So one way that antibiotics are oftentimes resisted by bacteria is that the bacteria will simply regulate its efflux pump and just pump the antibiotics out of the cell. And so there's interest, for example, in using phages that can infect the bacterium through the efflux pump as a receptor. And so that puts the bacterium under pressure to either mutate or lose its efflux pump, which then resensitizes it or prevents it from becoming resistant to these antibiotics. And so that's just one example of how we could use phages in tandem with antibiotics to have a more effective therapy. But beyond this, it's appreciated that, antibiotics are great. Again, they're broad spectrum, they have been consistent over time. But you also don't want to carpet bomb your microbiome, right? That has a lot of collateral effects that aren't ideal. Like we would, in principle, just want to target the very specific strain or species that's causing, problems with our health. And phages are very specific. They tend to target a given strain or species of bacteria, which in some ways has made them more troublesome as a therapy, because you need something that, again, is going to reliably treat the infection. But there is interest in using this as a way to modulate the microbiome or specifically treat the infection without having all this collateral damage that can also be problematic for our health. And this is just one other point that I always like to make. Another reason to, I think, study this genetic conflict, beyond just how fascinating it can be, is that over and over again, it has given us transformative tools, not just for gene editing, but also for the molecular biology that we do every day, right? There's friction enzymes that we have in our freezer that we use for digesting DNA and linearizing DNA. These are bacterial-enating proteins. CRISPR-Cas systems, right? Those have been hugely influential for gene editing. And I remember when I was a grad student, and CRISPR-Cas systems were just all the rage, right? Because it used to take us years to make knockouts, and now we can do it in a matter of days or weeks. And the list goes on, right? Free-lock systems, T7 polymerase, all of these have come from phage and bacteria, and specifically and oftentimes the genetic conflict between them. And yet we really only scratched the surface of what exists out there in nature. And I think as we explore this diversity more, we're going to find new therapies, new technologies that are just as transformative as what came before it. So in this part, I'm going to talk about how phages overcome CRISPR-Cas systems. And I'll talk also about why I think this is more broadly relevant to bacterial physiology. So I mentioned to you that CRISPR-Cas systems have been revolutionary for gene editing. But fundamentally, these are adaptive immune systems that evolved in bacteria to halt phage infection and also to provide memory against phage. And the way that they typically work is that when a phage injects its genome into the cell, snippets of its genetic material can be incorporated as spacers into a CRISPR array. And this CRISPR array gets transcribed and processed into these CRISPR RNAs. And these CRISPR RNAs have a spacer that matches the phage genome. So what they'll do is they'll guide a Cas nuclease or a complex to matching sequences in the phage genome and enable cutting of that genome. And that's how they confer immunity. And of course, everyone knows and loves Cas9, right? But that's really only one type of CRISPR-Cas system. There have been 6 major types discovered to date, and these can be broadly grouped into 2 classes. So one class has multiple proteins that come together in a complex to mediate cleavage. And one example of this is the type 1 CRISPR-Cas system, which I'll talk about today. But you can also just have a single effector nuclease that, along with the CRISPR RNA, is sufficient for targeting. And Cas9 is an example of this. Cas12a, which again I'll talk about a lot today, is another. And just as bacteria have evolved different CRISPR-Cas systems and different types that can target phage, so phage have evolved mechanisms, including anti-CRISPR proteins, that can overcome these CRISPR-Cas systems. And there's a lot of different anti-CRISPRs out there. They have different mechanisms of action as we'll explore. But the way that they typically work is that they'll bind to the Cas protein, oftentimes by mimicking DNA, and prevent the Cas protein from binding to its double-stranded DNA target. And so early in my postdoctoral work, I was interested in finding anti-CRISPRs against Cas12a, the type 5 CRISPR-Cas system. And this was in part because Cas12a was this new CRISPR-Cas system. Not much was known about it, other than that it cuts double-stranded DNA, and it's useful for gene editing. And so my hope was that by finding anti-CRISPRs against it, we might get some insight into this evolutionary arms race, and maybe novel mechanisms of action that we hadn't seen before, but also potentially useful tools that can make CRISPR-Cas editing safer. Because anti-CRISPRs are, on top of just being, a little biased, they're very interesting, right? They're also useful, right? So what we've seen is anti-CRISPRs can, for example, be introduced after CRISPR-Cas editing has occurred and shut off the nuclease and make it safer. So there is interest in using these just to make gene editing safer for patients. In principle, if we want to use phage therapy, we have to understand how phages actually overcome bacterial defense systems. And this, I think, is largely unexplored and part of why phage therapy has been kind of hit or miss over history. So by understanding anti-CRISPRs, we can potentially arm our phages to be more effective. And then finally, for phage engineering, which again is useful both for the molecular biology, but also for phage therapy, you can actually use these anti-crispers as selectable markers. And so with all this in mind, I set out to find anti-crispers against Cas12a, but none had been discovered before. And anti-crispers don't really have a domain or a motif that makes it clear that that's what they are, right? So what we oftentimes do, and what we did in this case, is to use indirect bioinformatics strategies to find them. And one way that we can find anti-crispers is by looking for bacterial genomes where the CRISPR-Cas system appears to be inhibited. And one signature of CRISPR-Cas inhibition is that you'll find this Cas genes, CRISPR-Cas nucleases, and they're co-encoded with a spacer that matches a prophage in the same genome. That's actually problematic. It may not appear so, but it's problematic because what the spacer would, in principle, lead the nuclease to do is to cut the prophage, which is part of the bacterial genome. And bacteria don't tolerate double-stranded DNA breaks very well, right? So for this bacterium to be viable and for it to have a spacer that targets a prophage in its genome, suggests that it also has an inhibitor that prevents the nuclease from cutting. And oftentimes these inhibitors can be found on the prophage that is targeted, which of course makes sense. And so we found these prophage targeting genomes in Marcella loboculi for Cas12a. And so that was a good place to start. It said probably there are Cas12 anti-CRISPRs somewhere in this genome. But how do you actually find the genes? Right? So another bioinformatics strategy that we use takes advantage of the fact that anti-CRISPRs tend to be clustered genetically in operons with other anti-CRISPR genes or with anti-CRISPR associated genes. So what we can do is take a known anti-CRISPR or anti-CRISPR associated gene and blast it and find it in new phage genomes. And then we can look and see, are there any genes next to it that are, let's say, hypothetical proteins of unknown function, right? These then become candidates for anti-CRISPR that we can test for activity. And you can go on and on and on, right? This can actually, depending on what you like to do on a Friday night, this can actually be really fun. But around the time that I was looking for Cas12 anti-CRISPRs, two students in the lab, Jenny and Adair, were looking for anti-CRISPRs against the 1F CRISPR-Cas type, which is a completely unrelated type. And Jenny had found this anti-CRISPR called ACR1F11, which I know is a mouthful, but it stands for anti-CRISPR against the 1F CRISPR-Cas type, and it was the 11th anti-CRISPR of that type to be discovered. So Jenny found that ACR1F11 inhibits the 1F CRISPR-Cas system, and she found that it was present in all these different prophages in different bacterial species. And interestingly, she found it not only in Pseudomonas aeruginosa, which is where she was originally looking, but also in Marcella catarrhalis. And that was exciting to me because I was looking around in Marcella loboculi for Cas12 anti-CRISPRs. And so the hope was that we could use ACR1F11 to find new anti-CRISPRs, not only against the 1F system, but potentially against Cas12a as well. And so I took Jenny's ACR1F11 sequence and I blasted it, and I found it is in fact in prophages infecting Marcella catarrhalis. And it's next to all these genes of unknown function, right? So all these became candidates for anti-CRISPR activity. And to test these, I did a phage plaque assay, which I'm going to show you a lot of today, so be warned. The way that these generally work is that we take a bacterial lawn that expresses a CRISPR-Cas system of interest, such as the type 1F CRISPR-Cas system. And we plaque phage lysates onto that lawn, a serial dilution of phage. And if the phage is able to infect, it forms these clearances that looked like black spots, right? So if you see black spots, think phage. These are really just clearances in the lawn due to cell death. So if the phage is targeted by the CRISPR-Cas system, as you see here, the phage isn't able to replicate and the lawn remains intact. But if you co-express with an anti-CRISPR, then the phage is able to replicate again because the CRISPR-Cas system is inhibited. So what I found is that this gene here inhibited the 1F CRISPR-Cas system, and so we called it ACR1F13. And that made me really excited about these genes in the middle, because they're flanked by two bona fide anti-CRISPRs and they're unaccounted for. So I looked to see if either of these genes were present in prophages in these prophage targeting Cas12a strains of Marcella loboculi that I told you about before. And what I found is that this gene here was in fact present in one of these prophages. And again, it was next to all these genes of unknown function that appear to be in the same operon. So I tested these to see if any of these could inhibit Cas12a. And I was very happy to see that 3 of them did. Like, this was one of those moments where you come to the lab on a Saturday and you get the result you were hoping for. It was just like such a great Saturday. But what we saw is that 3 of these genes inhibit Cas12 to varying degrees, as you see here. But that left one of these genes, again, unaccounted for. Right? It didn't look to be like any kind of Cas12a inhibitor. And so I thought maybe it inhibits another CRISPR-Cas system in this bacterium. Because as it turns out, bacteria can actually have multiple CRISPR-Cas systems. And the Morcella genus is actually very CRISPR-rich. So this Morcella voculi strain, for example, has the type 1 system, it has the type 3 system, and also the type 5. So half of the known CRISPR-Cas types. And in particular, it encodes the 1C system. And so I tested to see if this gene could be an inhibitor of the 1C system, and I found that in fact it is. It's actually very strong. And what was surprising is that this gene right next to it, which is a weak Cas12a anti-CRISPR, this also inhibited the 1C system. And that was really surprising because the 1C and the type 5 are completely different. There's almost nothing that is similar between them other than the CRISPR-Cas proteins with CRISPR RNA. But I looked around to see, again, where is this gene? Where else is it in Morcella and in other bacteria? And I found, again, it's co-encoded sometimes against genes of unknown function, which, of course, then I tested for anti-CRISPR activity. And what I found is that this gene here is also an anti-CRISPR, and it inhibits, again, both the type 5 and the 1C system, similar to how A0583 does, except that A0586 is a really strong inhibitor of both. And again, that was really surprising to us because this protein is less than 10 kilodaltons, right? It's a small protein, and yet it's able to inhibit somehow two CRISPR-Cas types that are completely unrelated. And so this is something that Jess in my lab is following up on to figure out how this small phage protein is able to do so many jobs so well. But this really just goes to show how this arms race goes, right? So bacteria evolve CRISPR-Cas systems to target phage. Phage evolve anti-CRISPRs to inhibit the CRISPR-Cas system. This probably leads to diversification of CRISPR-Cas systems, which leads to diversification of anti-CRISPRs. And even sometimes you'll get these anti-CRISPRs that have dual activities, right? But what I want to talk about for this next part is how really exploring these anti-CRISPRs and exploring this genetic conflict more broadly can uncover novel mechanisms of action that I think are more broadly relevant to bacterial physiology. So I mentioned to you that in this Morcella voculi prophage, we found multiple inhibitors of Cas12a. And one of these, called ACR5A1, inhibits Cas12 by cleaving its CRISPR RNA. And ACR5A1 was co-discovered and co-reported by the Down lab. And they went on to figure out its mechanism, and as it turns out, it's a great anti-CRISPR. It's robust, it's broad spectrum, it can inhibit diverse homologs of Cas12 that are divergent in the sequence. It's stoichiometric, it's enzymatic. It's really just a gangbusters anti-CRISPR. But that left the question as to why this phage bothers encoding multiple anti-CRISPRs against the same target. And it's not a trivial question, because phages are under strong selective pressure to minimize their genome. And this other anti-CRISPR, ACR5A2, which is what I'll talk about for most of the rest of this talk, it's a big anti-CRISPR. It's actually the biggest discovered to date. So normally these are under 10 kilodaltons, and ACR5A2 is over 37 kilodaltons. It's almost 1,000 base pairs. And so it's a mystery as to why this phage encodes this second anti-CRISPR when it already has one small one that's so good at its job. And also what this anti-CRISPR was doing. So I set out to figure out what its mechanism could be. But it so often is the case with these anti-CRISPRs, it doesn't really give us any clues. Right, so you can take this sequence, you can put it through any domain prediction software that you like, and it doesn't give you anything back, right? There's nothing that we really recognize. And even the crystal structure was solved a few years ago, and you can put that through Dolly, and likewise, it doesn't have any structural folds that really give us mechanistic insight. But there were some clues early on that it's unusual. So if we look at the distribution of this anti-CRISPR, what we see is that it's actually very widespread. So we see it in, we found thousands of homologs so far. And they're found in many different species and classes of bacteria. And what's surprising about it is that if we look at the distribution of Cas12a, what we see is that Cas12a very rarely coincides with it. So Cas12a is here in blue. You see that it almost never overlaps with the anti-CRISPR that's typically present on a phage or plasmid in the host. And that's really weird, right? Because anti-CRISPRs are usually very specific for their CRISPR-Cas system, so you wouldn't expect them to have such a little overlap. Then on top of that, what we found is that this anti-CRISPR doesn't seem to inhibit Cas12a directly in vitro either, the way that most anti-CRISPRs would. So most anti-CRISPRs, again, they'll directly bind to the Cas nuclease and block it from binding to its DNA target. But when we programmed Cas12a to target double-stranded DNA, which is what you see here, what we see is that anti-CRISPRs like ACR581 are able to block this, right? You can add an ACR581 protein, and this prevents the double-stranded DNA cutting that you see here. But when we added ACR582, there was no effect. It was very good at inhibiting Cas12a in bacteria, but not in vitro. And so that told us that there's some other mechanism of action. So I set out to figure out what this mechanism could be. And for a lot of the experiments I'm going to tell you about today, we're expressing Cas12a, one of two different orthologs, either from Moraxella voculi Cas12 or lactose-free CA bacterium Cas12. And we're expressing these from the chromosome off different non-native inducible promoters. And we're expressing the anti-CRISPR plasmid. And full confession, so we typically do this in Pseudomonas aeruginosa or E. coli, mostly because those are genetically tractable and Moraxella voculi is not. But what we see is that in this setup, this anti-CRISPR is able to inhibit divergent orthologs of Cas12a quite well. So even though INB-Cas12 and LB-Cas12 have only about 35% amino acid identity between them, this anti-CRISPR can inhibit both strongly. And we think that this anti-CRISPR inhibits Cas12 by downregulating its expression. Because when we look at Cas12a protein, we see that that's downregulated by the anti-CRISPR. And likewise, if we look at Cas12 mRNA, we see that that's also downregulated by the anti-CRISPR. And this particular result was really surprising to me because of what I told you about before. Right, so we're expressing Cas12 of different non-native inducible promoters, right? So we don't think that this anti-CRISPR is acting as a transcription factor because it's independent of the promoter, right? And what we found is that this anti-CRISPR could downregulate Cas12 mRNA, whether it was the native sequence or whether it was a codon-modified sequence, meaning that we could change the nucleotide sequence of Cas12 in the wobble position of the codon without affecting the protein sequence, and the anti-CRISPR could downregulate it just fine, right? And that was really surprising. Right? How does an anti-CRISPR downregulate the mRNA of a gene when both the promoter is changed and the nucleotide sequence has been changed? And I just want to emphasize that this is specific, right? So we did RNA-seq on these different strains to see if there were any other genes that were perturbed by the anti-CRISPR. And what we see is that Cas12a is the only gene that was significantly and substantially downregulated. So it is specific. And I scratched my head trying to figure out how this anti-CRISPR could do this. And I eventually came to the hypothesis that what it's actually recognizing is the polypeptide sequence. Because that's what's conserved when you change the promoter and you change the nucleotide sequence. And even though INB-Cas12 and LB-Cas12 are pretty divergent, there are nonetheless some amino acids that are conserved between them. And so I wanted to figure out how this anti-CRISPR could bind to a polypeptide and trigger mRNA destruction. But again, it doesn't have any nuclease motifs or anything that we can really identify. But it does have a lot of homologs. So what we could do is figure out which amino acids are conserved in the anti-CRISPR that are likely important for its function and then test them for their role in inhibition. And what we found is that there are amino acids in the C-terminal end of the anti-CRISPR that are very conserved. And if we mutate these individually to alanine, what we see is that the anti-CRISPR doesn't inhibit Cas12a very well, unlike what we see with the wild-type anti-CRISPR. And if we look at Cas12a protein, we see that that's also no longer downregulated, at least not very much, by these anti-CRISPR mutants, which correlates well with what we're seeing in terms of inhibition. And this was insightful because it linked the loss of inhibition to the loss of downregulation. But it was also really useful because one of the parts of our hypothesis that we wanted to test is there a protein-protein interaction taking place between the anti-CRISPR and Cas12? And normally that's a difficult thing to test because if you co-express them, Cas12a disappears, right? But what we could do is pull down instead on these anti-CRISPR mutants that don't fully downregulate Cas12 and then see does Cas12 interact. And in fact, it does. We saw that Cas12a protein came down with these anti-CRISPR mutants, showing that there is a protein-protein interaction that takes place. And so we wanted to know which region of Cas12a this anti-CRISPR is recognizing. And in Cas12a is about 1,400 amino acids, but I truncated it down from the C-terminal end to these smaller fragments. And what you can see is that if you truncate Cas12a to really just the first 30 amino acids, you don't see much downregulation happening. But as you increase to 100 amino acids and then 299 amino acids, you see that this downregulation does increase. And if you get up to 299 amino acids, which is really less than a quarter of the protein, we see pretty full downregulation happening at the mRNA level. But what I want to point out is that even though the first 30 amino acids of Cas12, which again is just the very terminal end, even though this isn't sufficient for downregulation, we found that it was sufficient for binding. Because if we pull down on this anti-CRISPR mutant, we found that the first 30 amino acids were able to come down with it. So that told us there's something within this very N-terminal end of Cas12 that this anti-CRISPR is recognizing. And so I made alignments of different susceptible Cas12 homologs to see what was conserved between them, right? What could this anti-CRISPR be recognizing? And I found that there are several amino acids that are conserved, but in particular, this LSKT motif. And as it turns out, this LSKT motif is conserved because it's important for Cas12a function. So this is looking at bacterial growth during phage infection, right? And what you can see is that when bacteria lack Cas12, they for the most part just get wiped out by phage. But if you arm the bacteria with Cas12, you see that the bacteria are able to grow, they're able to fight off the phage. But if you arm it with this mutated Cas12, it's sort of an intermediate effect. It's not as bad as not having Cas12a at all, but clearly the bacteria are compromised, and Cas12a isn't as effective as the wild type sequence. So that suggests that these amino acids are conserved because they're important for targeting, which of course makes sense. But we wanted to know, is this actually important for the anti-CRISPR? Is it important for recognition by the anti-CRISPR? And so we did another binding assay in which we looked at the ability of this anti-CRISPR mutant to pull down either wild type or mutated Cas12. And what we see is that, again, the anti-CRISPR can pull down wild type Cas12a just fine. But if Cas12a is mutated in these important amino acids, it's not able to pull it down anymore. So that told us that these amino acids in Cas12 are important for this protein-protein interaction that we see. But what about downregulation, right? So, so we again looked at the effect of these Cas12 mutations on downregulation by the anti-CRISPR. And we found that, again, just as the Cas12a is not bound by the anti-CRISPR anymore, likewise, it's not downregulated by the anti-CRISPR anymore. And this held even when we just made 2 amino acids in the Cas12a sequence. And so that told us that these amino acids are important for the binding by the anti-CRISPR and also the downregulation. And this happened, we saw this, this effect at the protein level, but also at the mRNA level. So when Cas12 is, again, mutated in these amino acids, we don't see this mRNA downregulation happening either. And I just want to emphasize that this really depends on changing the amino acid sequence. If we create silent mutations in the nucleotide sequence that don't affect the amino acid, the anti-CRISPR downregulates just fine. So we really think it's the polypeptide that the anti-CRISPR is binding rather than the mRNA sequence directly. But what about inhibition? Right? That's ultimately what the phage cares about, is whether or not Cas12a is able to target whether this anti-CRISPR still works. So we did another phage plaque assay. And what we found is that when Cas12a is mutated in these amino acids, basically the anti-CRISPR doesn't have much of an effect against it anymore, unlike what we see, again, for the wild type Cas12a. And this, again, is true if you change just 2 amino acids in Cas12. So you might then think, well, if these 2 amino acids are so problematic, right, why doesn't Cas12a just lose them? But it again goes back to that important role that these amino acids have for Cas12a function. So what we see is that if Cas12a is mutated in these 2 amino acids, again, it's not able to target phage very well anymore. So what we think is that this anti-CRISPR evolved specifically to recognize amino acids that Cas12a can't afford to lose. And that's what makes it an effective anti-CRISPR mechanism. But just to summarize our model as we see it, we think that this anti-CRISPR binds to the polypeptide of Cas12 and then triggers destruction of its mRNA. And all the data that we have suggests that it's co-translational. I don't really have any way of reconciling the data otherwise. And we do have a collaboration with your Vogels lab where they do see that the anti-CRISPR is enriched in polysomal fractions, which I think is consistent with this. But what I think is interesting about this is, we've never seen anything like this in either for anti-CRISPRs or at least to my knowledge in bacteria more broadly. But as a mechanism for molecular conflict, I think it makes actually a lot of sense. Because what this anti-CRISPR does is it recognizes amino acids in Cas12 that are broadly conserved in otherwise divergent sequences, and that again, Cas12a can't afford to lose. And so it has a way of downregulating divergent Cas12 sequences and also constraining the ability of Cas12a to escape it. And that's just a great way to antagonize your enemy and then downregulate it before it's even made in the first place. But I want to go back to this question that I proposed earlier, which is, why does this phage bother encoding multiple anti-CRISPRs against the same target? And in all fairness, I still don't really have the answer to this. We don't know for sure. But based on what we do know, I think we can hazard a guess, right? So what we see is that this anti-CRISPR ACR5A1 inhibits pre-existing Cas12 complexes by cleaving the CRISPR RNA. Whereas ACR5A2 will recognize the Cas12 polypeptide and then trigger its mRNA destruction, which basically interrupts its biogenesis. And I suspect that this is well suited for temperate phages that can integrate into the bacterial genome as a prophage and persist. Because this ACR5A1 will allow the phage to successfully infect, and then this ACR5A2 will allow it to persist in the genome, right? And decrease the chances that the phage and by extension the bacterial genome will be cut by Cas12a. So I think it's a way to actually maintain the symbiotic relationship between the phage and its bacterial host. But of course, there are a lot of questions that this project raised that we still don't know the answers to. I'd say the biggest one right now is what happens to the mRNA, right? So we have some idea of the mechanism of recognition, but not really what happens to the mRNA. But in principle, there's many things that could happen, right? So the anti-CRISPR could directly cleave the mRNA or recruit some other factor that does. It could instead cause ribosome stalling, or it could actually act on transcription. Because in gram-negative bacteria, transcription and translation are coupled, and everything we're talking about today is gram-negative. And so it's possible that this anti-CRISPR could bind to the polypeptide as it emerges from the ribosome, and then actually affect transcription as it's progressing. And even though I paint these as different possibilities, the truth is that they're all interrelated, right? So if you cause cleavage of the mRNA, that can lead to ribosome stalling, which can also then contribute to premature transcriptional termination. So they're all interrelated. But my hope is that we might get some idea of the proximate mechanism of how this anti-CRISPR functions. But we at least have some evidence so far that this anti-CRISPR doesn't act alone. Because if we take a very simple in vitro assays, where we have just Cas12 mRNA, and we add in a purified anti-CRISPR protein, plus or minus Cas12a protein and trans, what we see is that the anti-CRISPR doesn't act on Cas12 mRNA directly, which makes sense based on everything I've shown you today. But interestingly, if we use a slightly more complex system, a transcription translation assay that has only the bare minimum components that you need for transcription and translation, we see that the anti-CRISPR also doesn't downregulate Cas12. And I suspect that's because there are other factors that are present in the cell that simply aren't present in these very simple in vitro assays. And so we're interested in finding what these other factors could be, and we're using genetic screens and proximity bias simulation, these sort of approaches to find them. But in the meantime, Jorge in my lab is trying to rule out the usual suspects, right? So I mentioned to you that this anti-CRISPR could actually act with premature transcriptional termination. And there's a factor in bacteria called Rho terminase that is typically involved in premature transcriptional termination. And so what we did was inhibit Rho terminase using bicyclomycin to see whether or not this would restore Cas12a. And we see that it doesn't really have any effect, right? It kind of looks the same with or without bicyclomycin. So we suspect that Rho terminase isn't involved. And again, we're going through the list of other suspects that could be. But regardless of what is actually truncating or cleaving the Cas12 mRNA, it likely will converge on the same outcome, which is that you have basically a fragmented Cas12 mRNA and the ribosome is translating it. And it just gets to the end and there's no stop codon to release it. And these happen naturally in bacteria. Just things go awry and it can happen even without having a dedicated anti-CRISPR or anything like this. And so bacteria have evolved a pathway called translation that can resolve this issue, that can rescue the ribosome that's stalled at the end of the truncated mRNA and then clean up the mRNA and also the polypeptide. And I always like to talk about this because I think it's such an elegant mechanism. But the way that translation works is that you have this complex called the transfer messenger RNA. And this transfer messenger RNA, or tmRNA, is so called because it basically acts as a transfer RNA, but also as a messenger RNA. So it'll come into the unoccupied A site of the ribosome that's stalled at the end of this mRNA fragment, and basically just pick up where translation left off. So it has an alanine that picks up the polypeptide and will resume translation. And it'll resume it based on its own messenger RNA, which has an open reading frame. And this tmRNA will just add amino acids to this polypeptide chain that will allow translation to complete and will release the polypeptide that will release the ribosome. But it actually adds a sequence to the polypeptide that will tag this polypeptide for degradation by proteases in the cell. So it's just, again, a really elegant mechanism that will degrade the polypeptide, rescue the ribosome, and then the RNA also gets released and degraded by RNases in the cell. So we wanted to know whether or not tmRNA is involved in this cascade regulation, which it probably is. And also to delete the genes for the tmRNA to see whether or not we could capture any cascade fragments, mRNA fragments that would otherwise be rapidly degraded through translation. And so we used a genetic knockout strain where the genes for tmRNA are gone. And then we co-expressed with Cas12a and the anti-CRISPR. And what we found, I think quite surprisingly, is that when the tmRNA genes are missing and you co-express the anti-CRISPR Cas12, the anti-CRISPR doesn't regulate Cas12 anymore. So we see full-length Cas12a come back. And that was surprising given what we know about tmRNA, right? That it acts on truncated mRNAs. And so, you know, the interpretation at first blush would be that tmRNA is required for full-length Cas12a downregulation. But what I actually suspect is that expressing the anti-CRISPR and Cas12 in a genetic knockout strain that lacks genes to rescue the mess that creates is so toxic for the cell that it basically selects for mutants where Cas12a just doesn't get downregulated in the first place. So we're following up on this. I'm really hoping that there's just like a gene or something that's disrupted that might tell us what else could be involved in this downregulation pathway. But hopefully we'll get some insight to this down the road. But what I want to talk about before I finish up with this part is whether or not this is more broadly relevant for bacterial physiology. So I like anti-CRISPRs, and I was excited that this ended up being such an unusual anti-CRISPR mechanism. But does this matter beyond Cas12 and anti-CRISPRs? And I think the answer is yes, because of what I told you about before, which is that when we look at the distribution of this anti-CRISPR, we find that it's present on different plasmids and phages across many different species and classes of bacteria. And yet it very rarely coincides with Cas12a. So whatever it does in more cells look like to inhibit and downregulate Cas12, it probably isn't doing that in other bacteria. And in fact, this anti-CRISPR was actually discovered decades ago. It's a good lesson to review your literature, even from way back when. So this anti-CRISPR was previously reported as FinQ, which stands for fertility inhibition Q protein. And what they found is that this FinQ protein basically downregulates mRNA, but it downregulates the mRNA of transfer genes on F plasmid. Right, so when F plasmid spreads through conjugation from one cell to the next, it'll express these transfer genes that help form this mating hilus, and this is part of what helps the plasmid spread. And so what they found is that FinQ on one type of plasmid would basically downregulate transfer genes on F plasmid and just block this conjugation from happening. And they saw that the transfer gene mRNA was downregulated, but they never really figured out how it was doing that. And if we look at the predictive structure of FinQ and the anti-CRISPR and overlap them, what we see is that the C-terminal end is remarkably conserved. And many of the amino acids in the anti-CRISPR that I told you are so important for anti-CRISPR activity and for Cas12 downregulation, those are also conserved in FinQ. And yet, if we look at the other end of the protein, sort of the N-terminal and the mid-region, what we find is that those are very divergent. And so my pet hypothesis right now is that FinQ also recognizes polypeptide sequences, probably of transfer genes, and that this protein has simply diverged over time to recognize and downregulate different targets. So that's my pet hypothesis. We'll see if it holds out. But it's all to say that I think that this mechanism of genetic conflict, I think, again, it makes sense. It's a way to broadly downregulate divergent sequences. And so I wouldn't be surprised if it does exist in other aspects of bacterial physiology that just simply have to be uncovered. But in my very last part, I want to talk a little bit more broadly about anti-phage defense systems. But I think this is a really exciting time to be in this field, in part because we've come to appreciate that bacteria actually have a lot of different defense systems against phage. For so long, we only knew about really restriction modification systems and a couple of order of infection systems. And just over the past few years, there have been over 100 anti-phage defense systems that have been discovered. And if we look at the distribution of them, what we find is that even closely related strains of E. coli or Pseudomonas can vary dramatically in what their defense system repertoire is. And what we find is that these defense systems, they tend to be encoded on mobile genetic elements that can spread throughout the bacterial population. And so it's thought that this provides a broader scope of pan-immunity, that bacteria can share the communal resource that helps bacteria survive another day against the onslaught of phage infection. And another thing that's really cool about these defense systems, these newly discovered ones, is that many of them are evolutionarily related to proteins that we have long known to be important for mammalian immunity. And the list of this goes on, but one I'll talk about today is cGAS. So the cGAS thing pathway has long been known to be important for antiviral immunity in mammals. And what the field has discovered is that, as it turns out, cGAS is broadly conserved across the tree of life. We see it in all sorts of different species, and it likely originated in bacteria. And even though the sequences are divergent, the structures are very well conserved, and their mechanism of action is let's say, the overall pattern of it, I'd say, is broadly conserved as well. So prokaryotic cGAS, for example, will somehow recognize a viral cue. This is still a controversial issue in the field. And then it'll activate a signaling cascade of cyclic oligonucleotides that activate an antiviral response, typically through abortive infection, which I told you about before. So usually it'll activate some sort of a cell death cascade that again kills the cell, but does so before the phage is able to complete its replication, and in doing so spares the broader population from infection. And what we appreciate for these prokaryotic cGAS is actually incredibly diverse. So sting is just one potential downstream factor. There's actually many that have been found so far, and I suspect many more that will be. But all these discoveries raise a couple of big questions that, again, we still don't really know the answer to as a field. So one is, how do bacteria sense this phage infection, right? These abortive infection systems, they're deadly, right? They come at a huge cost to bacterial fitness. And so they're under tight regulation, and you don't want to activate them unless A, the phage is actually infecting, and B, the phage is winning, right? Because it is such a drastic option. And so what we're trying to figure out is how do these defense systems in bacteria sense phage infection? And then also, how do phages overcome these multi-layered defenses? Because we know that they do. Even though bacteria have over 100 different anti-phage defense systems, phage can still drive up to 50% bacterial mortality in a given day, right? And that's something that I'm interested in, and I'm planning to focus a lot of the future research in my lab on Klebsiella pneumoniae, which is a commensal bacterium that just has a really unfortunate habit of ending up in the wrong place, which can have very dire consequences for humans because it's a big cause of pneumonia and hospital-acquired infections, meningitis, and so forth. And what's scary about Klebsiella is that it is multi-drug resistant oftentimes. Again, it's a hospital-acquired infection. And so there is interest in using phages in order to treat it. But again, we don't really know what happens beneath the bacterial surface. There's a lot of work being done on the capsule of Klebsiella, but not much is known about how they defend against phage and how phages can ultimately overcome these defenses. So another area of research in my lab that's starting to open up is trying to understand how phages overcome this. I expect that phages probably encode proteins just like anticrispers, and that's part of the story. So we're planning to screen phage proteins for interaction with different bacterial defense systems in Klebsiella, and then screen them in bacterial models of Klebsiella for their ability to either inhibit these defense systems or potentially even activate them. But with that, I'll finish up and just make several acknowledgements. So again, much of the research that I told you about today was from my postdoctoral work in Giovanni Denny's lab at UCSF. And many, it sort of takes a village to get a paper out. Many of the people on this slide contributed very meaningfully to this work. So I'd like to just give them credit for that, and also for Joe being such a great mentor to me over the years. I'd also like to just highlight my growing lab, which I really like my lab, and I'm really excited to have their enthusiasm. I'd like to thank the University of Pennsylvania for giving me a job so I can continue this research. And thank you so much for being here. I'm really happy to take any questions that you have.
Time for questions. And maybe question. Please. The online folks can hear you. Feel free to turn.
Yeah. Yeah, thank you. That was awesome. Super interesting. I was love learning about phage biology. My question is, so you mentioned that the anti-CRISPR 5.2 was a 35 kilodalton protein, much larger than any other discovered anti-CRISPR protein. Do you have any reason why it's so large and why it hasn't been optimized to be much smaller? If it's not the one that's actually processing a lot of these events, it's actually sort of like docking a lot of proteins that actually perform the anti-CRISPR function itself. Why is it so large?
Yeah, I mean, I wish I could give you a truly satisfying answer to that. But I'll give you my best guess, right? So what we see oftentimes, a lot of these proteins that antagonize another, multigenetic element or they antagonize bacterial host, they're often just co-opted from different kinds of multigenetic elements. And I think in the case of this anti-CRISPR, my guess is that this is something that more broadly acts in bacteria, maybe on plasmids, and it's used to, mostly it's used by plasmids to antagonize each other. And plasmids don't have the same restriction on size that phages do. It's much easier to find very, very large plasmids. So my guess is that this phage just acquired this protein, and again, it evolved over time to have this mechanism. But because the mechanism is complex, it involves binding a polypeptide sequence and making sure it's the right sequence, right? Like it probably has more contacts beyond the LSK2 motif. And then it also has to do the business of downregulating the mRNA. And it could, like, we don't know exactly how it does that. It could recruit other proteins it does. It could have a nuclease motif that we simply can't recognize based on the nuclease motifs that we know of. And then it simply can't minimize beyond, or maybe hasn't had enough time to minimize beyond what it currently does. Whereas most of the other anti-CRISPRs that we see, it just has to kind of gunk things up, right? You can be pretty small and bind to sort of a nook and cranny in Capsule Bay and just prevent it from doing what it's doing. But I think if you have a complex mechanism where you have to recognize this and you have to recruit this, or you have to cut this, that probably you just need more space. So my guess is it would either be that, or that it simply just acquired something from a plasmid and it hasn't quite been minimized yet over time.
Well, really fascinating. You kind of alluded to my question, what you were just saying, and so you showed that the LSKT motif is necessary, right? So if you mutate it, then is it sufficient? Can you take that sequence and put it on another to get degradation of that?
Yeah, so not quite. So the LSK2 motif is something like the first 20 amino acids in Cas12. And what we see is that if we fuse that to RFP, that's not able to enable that regulation. And I suspect it could be one of two things. So my first thought was, it needs to make more contacts along the Cas12 polypeptide. Like that's necessary, but it's not sufficient. And maybe there's a broader part of the polypeptide that the anti-CRISPR has to engage in order to activate its downregulation. So that's one possibility. The other possibility is that it just needs a longer target, right? So maybe we took the first 30 amino acids and fused that to RFP, and RFP is simply too small of a gene for whatever it is that this anti-CRISPR needs to do. So right now what we're doing is taking different regions of Cas12, so 30 amino acids, 46 amino acids, 299 amino acids, and fusing that to other nucleases, like Cas9. And partially this is to get a sense of, what is, like, probably it's not sufficient, just because we didn't see that for RFP. But if it just needs simply a longer target, then maybe fusing it, the short motif to Cas9 will suddenly enable downregulation that we just didn't see for RFP. But my guess is that it probably needs a larger, like either a longer target or more amino acids that make contacts and enable the activity. Yeah. Yeah. So I don't know, I'm really excited about that because there is one example of this in eukaryotes, and only one, where there's a factor called TTZ5 that regulates tubulin expression by binding to the nascent polypeptide and triggering its mRNA. And it's a eukaryotic mechanism, so of course none of the proteins are conserved in bacteria. But it's interesting because it really just needs have very short polypeptide at the beginning, and it could be that it's simply just different in bacteria.
Okay, just kind of thinking about that, there's no other legions of Kinesiol that are also fully served, because you know that was easy.
Yeah, so that, that truncation experiment, it went on for a while, in part because I wanted to be agnostic as to what it could be recognizing. But what we found is that if you lop off the first 30 amino acids of Cas12, the downregulation doesn't happen. But you really need those first 30 amino acids, right? But what's interesting is that we tried the flipping experiment, where instead of having Cas12 fused to RFP, we had RFP fused to Cas12, and the anti-CRISPR could still downregulate that. So it doesn't have to be at the N-terminal end, but I think it just happened to evolve to recognize residues that are at the N-terminal end. Which, if it is a co-translational mechanism, the way that we think about it, it makes sense.
How was this? I was just curious about your this, which is found in so many bacteria.
Yeah.
And seems to be cured in the plasmid. Is it known, are they all on plasmid?
Distribution, it's kind of a mix. So that polygenetic tree was tricky because we find this anti-CRISPR on a lot of things where it's difficult to tell what it is, right?
I
Like,
think most bacteria, they don't know whether they have plasmid.
yeah,
So they just sequence stuff.
yeah. Well, I mean, so if it's integrated into the genome, for example, it's kind of hard to tell. Is it an integrative conjugated element, the transposon? So yeah, it's interesting. We do find it is pretty abundantly present in plasmids and also found in phages. But it's kind of a mix, and probably is found on other kinds of multi-genetic elements as well, but we just had a hard time calling them with any confidence.
How similar are these sequences?
So it's about 20% identity. So what I will say about this anti-CRISPR is that if we look at the sequence identity, what we find in more cell loculi, which is where we know that it acts as an anti-CRISPR, that's pretty divergent from all the other FinQ-like sequences. So whether or not it happened to diverge and it just became an anti-CRISPR in the more cell loculi lineage, or what exactly is going on there, it's not entirely clear. But it does look to be only about 20% amino acid identity.
Can you try to play to see whether you can find the seed structure of some of these?
Yeah, well, we did that.
Protein sequences.
Yeah, so we align the sequences themselves, and then we align the structures, and that's where, again, we see this really strong conservation in the C-terminal end of the protein, which again, seems to be the business end that's really important for the mRNA regulation. But the N-terminal, the mid-region of the protein tends to be variable in size and also in its sequence. And so that's why my pet hypothesis right now is that the N-terminal end is what's diverged over time to recognize other targets, while the C-terminal end is involved in the mRNA downregulation, and however it does that, it tends to be more conserved.
Check and see if there's. So I have one more question. It's kind of a straightforward question that perhaps I've missed. If you take fully folded or in vitro translated target Cas12 and your anti-CRISPR protein, do they pull down, put those down together, or does the target get unfolded?
I'm trying to do that experiment right now, because we don't know for sure how this is happening. So the way it happens in my mind is that as Cas12a is being expressed, the anti-CRISPR binds to it, and it can't downregulate because it's, the meat is missing whatever it needs for the downregulation, so it just stays stuck to it, with on-off rates taken into account. But we don't actually know if you mix them together, whether or not there's still this internal interaction. We do actually see a post-translational binding that happens, but it seems like there's actually a secondary binding site elsewhere in Cas12a protein that we don't know the significance of. As far as we can tell, it's not important for the inhibition that we see, but probably it's not there for nothing. But yeah, right now what I'm trying to do is express these either apart or together in like an in vitro transcription translation assay, parts so that we can tease apart these different binding questions and all that. But yeah, we don't really know if it's co-translational when that happens or post-translational.
Thank you. All right, well, let's thank our speaker. Thank you all for coming. And I do have your parking passes for the. It's gonna shut everything down here, I think.
Right.
And if you hit stop share. Let's just see, okay, we'll do this. Good. Cool, Vincent, thank you.